Alright, in the first video we discussed juxtacrine signaling and we talked about cadherins and how contact-dependent signaling can facilitate differentiation and survival factors and also organization of the tissues. Now we're going to talk about paracrine factors. This is where one cell secretes a ligand that is bound by the receptor of a nearby cell, and that causes signaling. So basically one cell is inducing another cell to respond and change in a certain manner. So my first question for you is, how would you be able to test if a signaling is due to paracrine, therefore diffusible, or if it's needed to be juxtacrine? So here we have two tissues, two adjacent tissues, and this is actually the lens and the neural retina. We've been talking about this in class. So this has a communication, induction, and competency. And how would we determine if that has to be attached, if it's contact dependent, if it's juxtacrine signaling versus diffusible? How would you determine if this has to be in contact or if these signals are diffusible? What do you think? See what you can come up with. Well, there are a lot of ways, but one of the major ways is just to do some tissue embryo manipulation and basically separate the tissues and put a filter there. Basically, this filter would be small and will allow diffusible paracrine factors to get through, but is no longer allowing contact-dependent signaling. So if the cadherins were important here, then that contact is lost and signaling would be gone. So juxtacrine signaling can no longer function. But if this is paracrine signaling and the factors are released and diffusible, they're still going to make it through the filter and you'll still get signaling. So we can determine if it's paracrine signaling or not based on if those factors are able to diffuse across the filter, or if we've lost the signaling capacity by separating the tissues, forcing them to no longer be in contact. All right, and so that's kind of what we're talking about here. If the signaling still occurs, those must have been diffusible factors. It must have been paracrine signaling. And indeed, when we're talking about the lens and retina development, these are diffusible factors. That's paracrine signaling. The neural retina and the lens will communicate with each other via paracrine signals. All right, so what is the nature of these paracrine signals? Well, basically, in order to get signaling, you need two things. You need a ligand, right? This is our induction signal, and you need a receptor. That's our competency. In order to be able to bind to and respond to the ligand, the cell must have the appropriate receptors. And so when the ligand binds to the receptor, there is a change in the receptor. In this case, you can see that the receptor is dimerized by binding to the ligand. The ligand holds the two receptors together, and then that leads to phosphorylation of the intracellular region of these membrane receptors. Sometimes it can just change conformation, but either way, there is a difference inside the cell that responds when the ligand is bound outside the cell. And then we are going to get signaling cascade, so that's going to activate downstream signaling molecules, and eventually we'll get a response. All right, so three common parts. Ligand binds to the receptor changes conformation, or at least has some definite intracellular change that allows the inside of the cell to know that the ligand must be bound. This leads to an intracellular signaling cascade and ultimately a cellular response. All right? So what types of responses can you think of? What types of responses might be something a cell would do? Well, there are three major types of responses. The first one is probably the most important with regards to development, and that's altering gene expression. So basically this ligand, once it signals the receptor in the signaling cascade, if it leads to new transcription factors being active and therefore new combinations of genes being on, that's inducing differentiations. So if the cell changes its gene expression in response to this signal, that is a huge part of developmental biology and differentiation. If you've taken advanced cell biology with me, you know that gene expression is not the only way a cell is regulated. It can also change protein activation. For example, just phosphorylating this signaling cascade molecule didn't change gene expression but turned it on. So this is a way that proteins can actually be activated without actually changing gene expression. But for long-term changes, a la differentiation during development, usually we're talking about gene expression changes. For immediate changes, a la what we talk about a lot in cell biology, that's a post-translational modification of the protein. And also it can change the cytoskeleton or adhesion capacities. So this is actually really important for cell movement during gastrulation. If a cell is moving, your cytoskeleton is very dynamic and the cell is motoring along. If the cell is now in the right spot, adhering to the right tissue, now you're going to change the cytoskeleton to more of a stable structural shape, and also you're going to create those adhesions between cadherins and so forth. All right, so all of these are things that the cell can do in response. We also need to talk about threshold, and we talked a little bit about this in specification earlier this week, but what do you think affects the threshold at which a cell will respond to this signal? Well, there are two main factors that affect threshold. Right? And first of all, let's define threshold. Threshold means the amount of signaling ligand that you need to get a response. So if you have a low threshold, that means you don't need much signal for the cell to respond. That's a low threshold. Think about a low threshold of pain. There doesn't have to be much pain stimulation for you to feel it. If you have a high threshold of pain, there has to be a lot of pain stimulation to get a response. Right? So a low threshold to a signal means you don't need a lot of ligand to get a response. A high threshold to a signal means you need a lot of that signal flooding the cell in order to get a response. Right? Well, the two main things that affect this are the number of receptors. All right? So what do you think is going to happen? Is more receptors a higher threshold, or would more receptors be a lower threshold? What do you think, and why? All right, well, let's think about this a little bit. And I introduced this in my Bio 2010 class. But basically, if you think about it, the more receptors, you have a better chance to bind a ligand. All right, so if I have a ligand here, right? Now let's take a look here. Over there, in the cups, now they're a little bit hard to see, but I have my receptors. All right? And in order for a signal, the ligand has to bind to the receptor. So if this was a carnival game, think about it for a second. Over here I have one receptor, and here I have 4 receptors. If you had one shot to win $1 million to get this into one of those cups, are you aiming for the one with 1 cup or the one with 4? Well, if you're smart, you're going to aim for the one with 4 because you have a much better chance of that ligand finding one of these receptors if there's more receptors. So thus, a smaller amount or even the same amount of ligand has a better chance of creating a signal if there are more receptors. Therefore, the more receptors there are, the lower the threshold. You don't need as much ligand to get a signal if you have more receptors. So, in summary, if you have more receptors, there's a better chance for the ligand to bind. Thus, you need less ligand to cause a signal, thus meaning, by definition, that the threshold is lower. So fewer receptors gives you a higher threshold. More receptors gives you a lower threshold. Okay? More receptors, lower threshold. The other thing that affects this is affinity. So affinity is defined as how tightly the ligand binds to the receptor. So what do you think about affinity? Would tighter binding ligand to receptor cause a higher threshold or a lower threshold? Well, tighter binding is going to give you higher affinity. So by definition, higher affinity means tighter binding. And if you have higher affinity, it's more likely for a smaller amount of ligand to bind. Right? You don't need as much ligand to bind if you have a higher affinity. If they bind tightly, it's going to find it and bind it and not fall off, very simply. Right? So if you have a higher affinity, binds tightly, you have a lower threshold because now you need less signal to respond. So let's take our analogy of the cups here again. All right? So I have 2 different receptors. One has low affinity, this cup here, and one has high affinity, this cup here. Now, you might think that the bigger cup is actually going to be better. Well, remember, when we're talking about signaling, we're not just talking about binding, but we're talking about attachment. And so if this is low affinity, it might bind, but it falls off. That ligand falls off pretty quickly too. And when it falls off, it's no longer bound, no longer signaling. However, if there's high affinity, once that ligand binds, it's bound tightly, it doesn't fall off, and you're continuing to get signaling. So with higher affinity, tighter binding, you have a more constant signal. You don't need as much ligand to get a signal. Therefore, your threshold is lower. All right, so again, in summary, more receptors gives you a lower threshold. You don't need as much ligand. Higher affinity also gives you a lower threshold. You don't need as much ligand. So let's talk a little bit about common signaling pathways now and how we can look at these. So here's a common factor, fibroblast growth factor, receptor tyrosine kinase, FGF-RTK signaling pathway. So the ligand is an FGF, the receptor is a receptor tyrosine kinase, and then you have all these signaling cascade molecules. So when the ligand is bound, the receptor will phosphorylate itself. It's dimerizing and the two molecules, the two receptors are dimerizing and phosphorylating each other, and that leads to the adapter protein now binding because it wasn't going to bind without the phosphate group. That leads to activation of a RAS GDP to RAS GTP, which then activates RAF, activates MEK, activates ERK, causes a transcription factor to now become active, and now you've altered your gene expression. And if we just see this here, arrows just mean activation. So when the receptor tyrosine kinase receptor is bound, it activates GNRP, activates RAS right here, activates RAF, MEK, ERK, and so forth. And so you can imagine that this whole cascade is needed. If any of these parts of this cascade are disrupted, for example, if RAF is mutated, then you might have ligand activating GNRP, activating RAS, but RAF being mutated, you've blocked the signal, you're not going to get transcription. So anything non-functional along the way here will lose transcription. Right? And so here's an example of that. During development, there are a set of cells called the neural crest cells that we'll also learn a lot about later on. So these have to migrate from the central nervous system, basically migrate from the detachment point of your neural tube from the epidermis. They are going to migrate out towards the periphery. And when they migrate out towards the periphery, some of these cells become melanocytes. These are pigment cells. And the melanocytes are going to express melanin, and so if you stain for melanin, you can see these melanocytes here, highly stained for melanin, lots of melanin being produced, hence giving your skin pigmentation and so forth. Right? We actually saw this already because we know that in our chimera transplantation experiments, when we took transplantation from the neural tube, and we transplanted it into the host. So we took pigmented cells from a quail, transplanted them into non-pigmented chick. Well, indeed, the quail cells were in the neural tube where you'd expect, but they also migrated. Those were the neural crest cells that were derived from this that migrated out, became ganglial cells, sensory neurons, and we see here M. M stands for melanocytes. So that migration is really important. Now, what do you think would happen if any of this was mutated? So some of those cells become melanocytes because of signaling that causes differentiation, and that signaling is through an RTK receptor called KIT. And if KIT is mutated and non-functional, what do you think is going to happen? Well, we'll see that in a second. Also, MID-F is the transcription factor, or the transcription factor for our melanocytes. Remember we said it turns on a transcription factor. Go back here, way back I guess. Turns on a transcription factor. Well, different family members of FGFs and different receptors will turn on different transcription factors, but one of them is MID-F. And so when MID-F is turned on, activated, that's what causes the melanocytes to differentiate. And if you have non-functional MID-F, this is what your staining looks like. So now we're staining for melanin. So what do you think the phenotype of these mice that have a non-functional MID-F would be? Well, these are basically albino mice. So if you've ever worked with mice, you know that there are mice that are black, mice that are brown, and then there are white mice. And the white mice are a very common lab mice, but the reason they are albino white mice is because of a change in MIT-F. And so the melanocytes are no longer able to differentiate because even though they're getting the signal receptor activated, they're not changing their gene expression. They're not getting the result of the signal. My next question for you is, would you predict this genetic mutation for these mice to be in the gene for MID-F, the enhancer for MID-F, or the enhancer to which MID-F binds in order to activate pigmentation? Right, well, the answer can actually either be B or C, but it would not likely be A. All right, so let's have a follow-up question now. I've given you the answer and we'll talk more about why these two would work, but my next question for you is why would it not be A? Why would the gene for MIDF, the coding region of that gene, not likely be the mutation? Well, the reason for that is because if you mutate the gene for MIDF, you have a non-functional MIDF protein, and MIDF is probably a transcription factor involved in a lot of different gene transcription regulation. So remember, we're talking about combinatorial enhancers. Right? MIDF in combination with several other things is needed to turn on this gene for melanin and differentiation into melanocytes. So if MIDF's not there, you don't have the right combination. Even if you should, the melanocytes no longer differentiate, albinism happens. Right? The mice become albino. However, if you don't have MIDF anywhere, it's one thing to not have MIDF in these particular cells. It's another thing to not have MIDF at all. And now every gene that requires MIDF as part of the transcription cascade, or sorry, as part of the transcription factor combination to turn it on, all those genes are non-functional. That would be lethal, right? And if MIDF is in a repressor, well, genes that aren't supposed to be on will be, and that also is lethal. So because MIDF probably has so many different functions in turning on and regulating so many different genes, if the gene for MIDF itself is mutated, that would be lethal. However, if the enhancer for MIDF is mutated, that won't be lethal. Now, we maybe just do not have MIDF expressed in these cells, but it still might be expressed elsewhere, and it can still turn on other genes. We've only affected the differentiation of the melanocytes because MIDF itself probably has multiple modular enhancers. And so if the enhancer for MIDF that turns it on in the melanocytes is the only one affected, MIDF is still expressed other places, and turning on other genes, only your melanocytes are affected. Also, the enhancer to which MIDF binds. So if the enhancer for melanin, or the enhancer that causes differentiation into melanocytes is mutated, well now MIDF might even be active, but it can't bind to that enhancer anymore, and now it can't turn on that. That is only going to affect the gene for melanin. That could also cause albino to occur. All right? So it can't be the gene itself because MIDF has so many functions. Right? Pleiotropic has a lot of functions. But it could be the enhancer for MIDF because the modular enhancers, you affect one spot, it's not going to affect everything else. Or if MIDF is even active, but the enhancer for the melanocytes now is changed and the sequence that MIDF binds to is no longer there, well now MIDF can't activate that and the melanocytes will be affected. But either way, with B and C, we are just affecting the melanocytes. The development elsewhere would be allowed. All right, so that's a hugely important concept, so hopefully that's making sense there. All right, so, but you can imagine that if we have MIDF not working, that can lead to albinoism. Well, if the KIT receptor is also not working, that would also lead to albinoism because ultimately you still lost the same signaling pathway. And the ligand steal, the particular ligand for this, could also be mutated. And we've seen this as well. So when either steal or KIT are mutated, that leads to piebaldism that we've seen in humans and in mice. And this is because the neural crest cells that migrate out and form pigment kind of in the trunk, the belly region, and in part of the head, those are no longer able to differentiate. And so you get, in this case, just lack of pigment in those regions. But remember, we have not affected the whole gene itself. And so you still get pigmentation elsewhere. Another common signaling pathway is jackstat. All right? So what I want you to do is kind of look at this briefly and try to write down just in 3 steps what's going on. All right? Can you identify the important aspects of this pathway and write down what's going on? Because this is really what I want you to be able to do. You don't have to memorize any pathways, but you have to be able to look at a pathway given to you and kind of determine what's going on. So what's happening in step 1? Well, in step 1, prolactin is our ligand, and it's binding to our receptor and causing dimerization. And upon dimerization, what happens? Well, upon dimerization, the receptor phosphorylates each other. Jack, which is bound to it, now becomes phosphorylated as well. So once Jack becomes phosphorylated, it now phosphorylates stat, and when stat5 is phosphorylated, or there's a lot of different types of stats, that causes dimerization, and the dimer is able to get into the nucleus and bind to the enhancer region and basically regulate transcription at this point. So my next question for you is, what is this? All right, so it says casein gene promoter. So the gene for casein, that's just a gene. But this whole thing wouldn't be in the promoter. The promoter would be down here with the TATA binding protein and so forth. And we can see these kind of double lines here that indicate there's a lot of distance between these. So what is the part that this is actually binding to? Well, that's the enhancer region. So the transcription factor in combination with another transcription factor, GR in this case, binds and causes expression of this gene. So an example of defective STAT pathway. So this is a mutation that can be passed down, and this is where the kinase domain, all right, so if we look at this kinase domain here, it's constitutively active. So it's not non-functional, but it's always phosphorylated. So what that means is it's always signaling jackstat, even if there's no ligand. And so this particular pathway with FGF receptor 3 is important for kind of promoting differentiation from cartilage into bone. And so you don't want the cartilage to turn into bone before it's done growing. And so when this is constitutively active, as soon as there's cartilage, it causes it to turn into bone. And this leads to a disease called thanatophoric dysplasia. And unfortunately what that means is basically the bones don't grow before they harden, and so the limbs and all the skeletal bones are very tiny in this infant. And so it's because the cartilage precursor cells basically turns into bones of the long bones. They stop cell division too early, they don't grow. And unfortunately, this leads to death when this child is born because the ribs are not large enough to allow the lungs to expand. So unfortunately, this is a lethal mutation caused by signaling cascade that's on when it should not be. All right, another common signaling pathway, the hedgehog family. All right, and so there's several hedgehog family members: desert hedgehog, Indian hedgehog, and then finally sonic hedgehog. All right, the fish, there's a couple of other ones as well: echidna hedgehog and tiggywinkle hedgehog. These are actually from Beatrix Potter books. So they ran out of actual hedgehog names and then started kind of making some up based on characters. But remember, genes often are named for what the phenotype looks like when they're knocked out. So here we see where sonic hedgehog, SHH, this is in situ hybridization, so it's where it's expressed. So based on that, what do you think it's involved in? Well, it's involved in development of the feathers. And so when sonic hedgehog isn't functional, when it's knocked out, you don't get feathers, and therefore they decided the embryo looks like a hedgehog because it has these bumps instead of feathers on its back. So it's named based on the phenotype when it's knocked out. But sonic hedgehog is another key signaling ligand, and so it binds to the receptor patched, and we can kind of look at what's going on here. So normally, in the absence of ligand receptor, pKa and SLIM are cleaving this Cy protein, cutting it in half. And when it's cut in half, that's a repressor, no transcription. However, when we have sonic hedgehog, when it's bound, now pKa and SLIM become inhibited. Cy is not cut in half, and now it's an activated transcription factor. You would get transcription. And so here we have what's called a double negative gate. Right? So patched is normally inhibiting smoothened, but smoothened can inhibit pKa and SLIM. And when hedgehog's here, it stops patched from inhibiting smoothened. And so basically hedgehog is now activating smoothened, and smoothened ultimately is going to result in Cy being active. But we can think about this another way. Hedgehog inhibits patched. Patched, in the absence of hedgehog, is actually inhibiting smoothened. So when we have no hedgehog, patched is inhibiting smoothened, preventing it from doing its job. Well, what is smoothened's job? Smoothened's job is to inhibit pKa and SLIM. pKa and SLIM's job normally is to inhibit the Cy protein, right? And the Cy protein's job is to activate transcription. So when we think about it, as soon as we have hedgehog, patched is no longer inhibiting smoothened. So hedgehog basically activates smoothened, kind of negative, negative, double negative gate, go around it. So smoothened is now active. And if smooth is active, it inhibits pKa and slim. And since pKa and slim is inhibited, it can no longer inhibit psi. So smoothened basically activates psi, and that leads to transcription. Okay. So my question for you, what would happen if, and so think about A, would it be constant transcription, transcription's always on, or no transcription, if pKa and slim was mutated so that they don't work? Well, if pKa and slim are mutated such that they don't work, well then you never have psi inhibited. Even if there's no hedgehog, pKa and slim are not cleaving psi. Psi is always active. You're always going to get transcription. If you don't have this inhibitor, there's no way to inhibit transcription. Transcription's always on. If smoothened is mutated such that it doesn't work, well now you can't inhibit pKa and slim. So now even if hedgehog's there, you're not inhibiting pKa and slim, and therefore pKa and slim are constantly inhibiting psi. You're not going to get transcription at all. So if you're on this kind of green line here where we do our double bypass of the double negative gate, if you're on the green line, that means you're activating transcription, and if you're mutated, you can't get transcription. If you're in between here, you're inhibitors, and if you're inhibited, then you always get transcription because it's no longer inhibited. All right, so we've talked about sonic hedgehog. Here we can see that it's pleiotropic. It's expressed in a lot of different things. It has a lot of different functions, including limb development, a little bit of brain development, neural tube development, and so forth. If sonic hedgehog is blocked, you get major mutations like we've talked about in lab. This is a lamb that ate a chemical called cyclophamine, or the mom ate the chemical called cyclophamine. Last pathway is Wnt. All right, so here we see another double negative gate. So let's think about what's happening now. Well, if we work from the back up, it becomes easiest. Beta-catenin causes transcription. GSK3, well, inhibits beta-catenin, therefore inhibiting transcription. What's disheveled's role? Well, this is our double negative gate. Disheveled inhibits the inhibitor. And if you inhibit GSK3, it no longer can inhibit beta-catenin, therefore disheveled is going to activate beta-catenin. And this is how Wnt ultimately leads to transcription. It activates frizzled, frizzled activates disheveled. Disheveled then, by inhibiting the inhibitor, allows beta-catenin to be active and causes transcription. So, what would happen if frizzled was mutated and non-functional? Well, if frizzled was mutated and non-functional, you could not get transcription because there would be a block in the pathway here. There's no way to activate disheveled, therefore GSK3 is never inhibited, and it is always inhibiting transcription. So if frizzled doesn't work, you're never going to get transcription. But what about if GSK3 was mutated and non-functional? Well, if GSK3 is mutated and non-functional, now you don't have the inhibitor. And so now, even if there's no Wnt, nothing is inhibiting beta-catenin. Beta-catenin is constantly going into the nucleus and causing transcription. So if GSK3 was inhibited, you lost your natural inhibitor as part of the double negative gate. Therefore, transcription's always on. And then finally, if beta-catenin's mutated, non-functional, well, in this case, you'd never get transcription. So basically, everything here, if you inhibit Wnt, frizzled, disheveled, beta-catenin, any of those, you're not going to get transcription. But if you inhibit GSK3, that one causes transcription all the time because that is your natural inhibitor keeping this pathway off until the Wnt signal causes it to be on. And we don't really need to understand the other pathways, but there's a non-canonical pathway to Wnt as well. Wnt is very involved in all kinds of signaling, gene transcription, activating transcription factors, etc. So these are just kind of very broad families that encompass a lot of different signaling. We have the FGF1, jackstat, we have hedgehog, and we have Wnt. Thank you. And again, you don't need to have any of those signaling pathway, kind of like we worked on the last 10 minutes. If I give you a signaling pathway, you should be able to kind of tell what's going on, and you should be able to tell me what's going to happen if this is mutated. And so if you've struggled with that a little bit, come see me. I'm more than happy to help you out with that.